Hello, everyone, and welcome to today's live broadcast, Actionable Insights Delivered Through the Seamless Workflow of the GeneReader NGS System, presented by John Leamon. I am Brenda Kelly-Kim of LabRoots, and I will be your moderator for today's presentation. Today's educational web seminar is presented by LabRoots, the leading scientific social networking site and provider of virtual events and webinars, advancing scientific collaboration and learning. We'd also like to acknowledge the C&EN Media Group for helping with programming and promotion of the event. This webinar is sponsored by QIAGEN Molecular Diagnostics. QIAGEN Molecular Diagnostics provides solutions from sample to result for companion diagnostic testing, oncology, infectious disease, and detection of sexually transmitted diseases and latent tuberculosis. See more at www.qiagen.com/US products molecular diagnostics. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many of your questions as we have time for at the end of our presentation. Also, please notice that you will be viewing the presentation in a slide window. To enlarge this window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window or use the Q&A button lower left to let us know you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your web page and follow the process to obtain your credits. I'd now like to introduce today's presenter, John Leamon. John Leamon is the Senior Director and Global Development Lead for NGS at QIAGEN. He has published 25 papers, sits on several scientific advisory boards, and is an inventor on numerous issued patents and patent applications. For more information on John, please click on his photo image at the top right. Please join me today in welcoming John Leamon. I will now turn it over to him for his presentation.
See. Perfect. Sorry about that. Sorry for the delay there. I missed the introduction, but I'm sure that it was excellent. So thank you, everybody. Appreciate everyone's time. And today I'd like to walk you through QIAGEN's offering for a sample to insight workflow. And I'd like to spend some time to take you not only through the basis of what forms our sample to insight workflow, and how we arrive at the content, which of course is the most critical part, but also then walk you through the workflow itself and give you some clarity into what that process entails and why we think that's going to be of interest to a wide number of users. So obviously I think everybody's pretty much of the opinion that NGS solutions are rapidly becoming more and more constant within the medical community, within the research community. And certainly with that, as it moves from the very basic research kind of venue to more of an applied field, there are a couple of challenges that have to be handled and have to be addressed before the adoption can become complete. So let's just break those down into four rough categories here. One of those, of course, is if we're going to discuss the use of targeted panels, the actual question of designing the panel itself. And so their questions are, for example, which genes should be included and why, which variants are significant and why. And typically, at least in my experience, this may come down to kind of a hit list of genes that are of particular interest within a particular lab, driven by perhaps the interests of the PI or postdocs or key opinion leaders. And then this is going to be kind of a constantly moving target because these are based rather than any real solid kind of actionable data, but more driven by the literature, which, as we all know, evolves. So that's a moving target, and that creates issues. Secondly, there's always the questions of the interpretation itself. NGS generates a large amount of data. It's one of the attractive features of it, obviously. But how do you handle that large amount of data? Not only from the interpretation side, but really from the data itself. Where do you store it? How do you store it? What do you do with all that data? But then, from the interpretation aspect, how do you go from this raw data to meaningful and eventually actionable results. There's a question, of course, of information overload. There is a lot of data that we generate. What do you do with numbers of these variants of uncertain significance that come through? What might a clinical researcher do with those? What actions do you take based on findings that maybe are less than certain but could potentially have real-world impact? And then lastly, there's certainly cost issues. All of that data that you generate can lead to unnecessary work, whether that's the bioinformatics analysis, and the interpretation questions, all of these issues, storage, are all resulting in higher costs, extra work. And then we also have to remember that those unnecessary testing is in effect costly to a laboratory and is going to be undermining various business cases that may be settled around that. So these are kind of four basic adoption challenges that need to be addressed. So to address those and to certainly ease the transition of NGS from a strictly research tool to one that's more applied, QIAGEN offers two big improvements on existing workflows. One is the use of actionable insights, where the reports that QIAGEN generates using our gene panels are proven and relate directly to actionable variants, ones that you can actually do something about. And secondly, and this will be the second part of the talk, we'll discuss a little bit about the workflow, how we have the first truly complete workflow for the NGS system, where you have one partner who provides an entire solution, not only from purified DNA, but really, as we'll show, from FFPE all the way through to the report, and all within one product. So that will be the second part, as I said. So that actionable insights, where we're creating the actionable reports. The interesting thing about this, which I find particularly unique, is that the whole basis of the gene panel that we use for these actionable insights stems from the QIAGEN knowledge base. And the knowledge base is a database that QIAGEN has generated. It's comprehensive, it's industry leading really, and as you can see here, it's a compilation of multiple different sources, both public and proprietary. And it's really, this has to be, can't be stressed heavily enough. It's expertly curated and continuously upgraded or updated to provide this kind of up-to-date knowledge. And so as you can see, you can form, the knowledge base takes the information from drug labels, from results of clinical trials, from external databases, professional guidelines, and then lastly, you have scientific literature, the clinical case counts, and those run specifically through that kind of expert curation and continuous review before they're added to the knowledge base itself. So that's the basic QIAGEN knowledge base. Now, how we use that to guide the panel design is kind of unique, because traditionally, I think people take their list of favorite genes or list of favorite variants, and you generate a panel of these, and then you go and try to determine what actions can you take, what do those variants mean. We took the reverse. We essentially stood the conventional wisdom on its head, and instead of starting kind of from the panel and trying to see which disease types we could apply it to, instead we took the tumor types, the 5 top tumor types in this case, and we then went to the knowledge base itself and looked for variants that were linked to medical actionability within those specific tumor types. And so as a result, we figured that we determined which amplicons, and then from those amplicons, what was the panel that would result in a necessary and sufficient list of variants that would give maximal actionability in clinical research for those top tumor types. So in this case, what we were doing is we took the tumor types, the 5 tumor types, and instead of using, and this is kind of important, we didn't use the entire knowledge base to inform this decision. Instead, we stuck with a rather conservative approach of only using data that came from drug labels, clinical trials, and the professional guidelines. Okay, we did not rely on the scientific literature, because that is more of an academic kind of pure research element. Extremely important for some people, absolutely, but in this case, we were looking for more conservative guidance, and we didn't use the external databases. So it was a reduced data set that went into applying those. But what we did, so again, I said that it was top 5 tumor types. And so in this case, we used breast, ovarian, colorectal, and lung. And we chose these 5 on the basis of the prevalence, the health impact, and critically, the availability of targeted therapies with proven genetic biomarkers within these 5 types. And so what came out, if I'm going to jump back really quickly, so you see kind of going into the query itself would be these 5 tumor types that go into the knowledge base. And then the result which comes out is these 12 genes, which are listed below: KRAS, NRAS, KIT, BRAF, PDGFRA, ALK, EGFR, ERBB2, PIK3CA, ERBB3, ESR, and RAF1. So those are the genes that came out as having the highest percentage of actionable insights, of actionable variants that would be linked to targeted therapies within those 5 tumor types. So we're going to just take a quick walk through the panel. The panel, those 12 genes obviously were covered by a specific panel. And so I'll take a quick walk through the panel itself and give you some insights into that. So the panel size itself, and then we'll actually this isn't just kind of sharing detail for detail's sake. We'll then compare it to some of the existing panels that are on the market and go through some of that information. So the panel size itself, we said it's 12 genes, only covers 16.7 kilobases of data. But from that, and this is important, there are 773 unique variant positions. Those variant positions are covered by 330 amplicons. Okay, so, and if we take a look at this, we'll see that they give us a variant frequency where the industry feels comfortable calling, which is 5%. We can actually go lower. We've got data that shows going significantly lower, and I think that we cover some of that in as we walk through some of the data that we went with the Broad. But obviously the kind of accepted cutoff is 5%. And what's really interesting about this is the 330 amplicons. You'll see that with the variant positions, there's obviously multiple variant positions within specific amplicons. But when we look at the coverage, and this is using just as kind of a test case using NA12878 DNA, but 12 different data sets, you'll see that of those amplicons, and these are the entire amplicons, you get 98.5% of those amplicons have a coverage of greater than 200x, and 96.4% of those amplicons have greater than coverage of 500x. So that's the amplicon. But again, what I mentioned was that we're specifically interested in the variant themselves. And so these amplicons are designed to maximize coverage of specific variants. And so when we look at that, the numbers are even more impressive. We're looking at 99.9% of the variants are covered at greater than 200x, and 99.8% are greater than 500x. So I think that should be an indication this is a well-designed panel. It covers the target variants extremely well. It covers under a very low number of amplicons. And let's see how that looks in comparison to what are the other 2 kind of major oncology-related panels that are out there. So again, this is just a distillation of our first of the GeneReader AIT panel, Actionable Insights Tumor panel, in the leftmost column. And then we compare that to the Illumina in the center and the AmpliSeq, Thermo Fisher's AmpliSeq panel on the far right side. And we'll see that of the panels that are there, the GeneReader panel is the smallest, not only in terms of genes that are covered, but also in the sheer amount of data that's provided, or genetic information that's covered. Fewer variants are covered here. We're on 773, concentrating again on these unique variant positions. The number isn't listed for the Illumina panel, but it's 2,800, almost 3,000 variants for the AmpliSeq. However, interestingly, you'll notice that again we cover the smaller amount by the largest number of amplicons, and so that's kind of is speaking again to the level of coverage that you can presumably expect. And all of these have the same basic level of variant frequency that they prescribe to, the 5% cutoff or above. However, when we look at the coverage, again, what we're going to show the AmpliSeq coverage here, or excuse me, the Actionable Insight Tumor panel here, and compare that to the other ones. You see that we've got 98.5% greater than 200x, average coverage of 5,000x for the Actionable Insight at the amplicon level. As opposed to 93.5% for the Illumina panel, and the numbers aren't actually given for the coverage for the AmpliSeq. However, we get greater than 500 or 5,000x coverage for the AIT panel on a per amplicon basis, whereas you get somewhere between 1,000 to 4,000 for the AmpliSeq. So what we're really seeing is that although the coverage, the number of genes is higher in the Illumina and the AmpliSeq panel, the level of coverage, as you might expect, is lower. And then we look at the variable insights, which because they aren't, the other two competitors aren't insight related, they don't have that data. But let's take a look at the actual genes that are covered. So there are 12 genes that AmpliSeq covers. Let's take a look at these and see what the level of coverage is within these. So this is just a general Venn diagram, and so in the very center of the Venn diagram, I'll take a step back. The blue circle are the genes that are in the QIAGEN Actionable Insights. The yellow is the Illumina, covered in the Illumina tumor panel, and the black is the Thermo AmpliSeq panel, the hotspot. And you'll see that there are 8 overlapping genes that are covered in all, that are common to all 3 of those panels. The Thermo Fisher, 42 genes covered in that aren't found in the other ones. We've got 17 genes that are in the tumor or the Illumina offering. And then interestingly, even though it's a low number of genes, the smallest number of genes overall, there are 4 genes that are unique to the QIAGEN panel that aren't covered in the other 2. So let's take a look at the genes. So even on those 8 genes that are covered, and they're listed on the column to the left, let's take a look at those genes that are covered in all of them. So we've already acknowledged that there are some genes that are covered in one panel, not the other. But let's take a look at the insights, kind of these actionable variants that you can find in the genes that are covered. And so if we take a look at this, this is the, in comparison to the coverage, the variants covered in the actionable insight, this is just a listing by gene of the number of actionable insights that are not covered in either of the 2 panels. So for example, for EGFR, there are 58 insights of variants that aren't covered by Thermo Fisher, 28 that aren't covered by Illumina, and then there are actually 21 of those that aren't covered by either panel but are found in the actionable insights. And so you can run through all the different genes, and at the end of the day, for these 8 that are in common, you find that there are 81 variants that aren't covered in either of those 2 panels but are found in the QIAGEN. So again, this just speaks to, I think, 2 things. One is the design criteria that was used going in, using the medical information, the clinical research information, the existing data that was out there to guide which variants we thought we should be investigating and designing panels specifically to cover those. But also the ability that we were able to really specifically target those variants and ensure that they were covered successfully. So now this is going a little deeper, and so in this case what we're going to do is we're going to take a look at an example of one of these variants that was found only in the QIAGEN panel design. I mean, I think the question obviously is raised, okay, so there are variants that are covered, that QIAGEN covers that other companies don't, but are those important variants? You know, they may not be all that important after all. So we'll just dig into one particular case. There are many others, but in this case, what we're going to look at is we're going to look at a variant that's found in ERB2. And you know, here it is outlined in the red box, and you can see that it's a missense mutation. So the first thing we find is that actually the question of whether it's relevant at all, it pops up in 27 different publications. So this is at the very basic kind of research level. One would assume that this is in fact a relevant mutation, as it's been covered fairly extensively in different mutations. So from that sign alone, perhaps it would be worth including. But if we actually begin to take a look at those publications, what are they covering? Are these just strictly research kind of academic interest, or these have some applied sensibilities? Certainly, we looked at there's Nature, there's Frontiers of Oncology, and we find that this mutation is an activating mutation and is actually discovered when you're looking at molecular profiling for adenocarcinomas in lung. So again, this seems to be increasingly relevant. And if you dig into the research a little more, you know, it's found at significant frequencies, and as I said, it's an activating mutation. So then, actually, if we dig in further, we find that some of those publications are indicating that it's actually associated with drug response. So again, one would make the claim that this is one that would be in almost anybody's list. And now if we look at, so we've established the fact that this seems to be a relevant mutation, one that's worth investigating. And now what we'll look at is the back end of the system. We'll get into this a little bit more when we talk about the workflow. But the QCI, the Ingenuity backbone of the bioinformatics software, is what really provides that linkage that connects the researcher from the data that they've generated using Actionable Insights tumor panel to the data, this kind of actionability, the relevance of what it is they're finding. So if this is an indication of the report that would be generated on the basis of identifying this particular variant. And first of all, what you get is obviously the classification, tumor content, the kind of the diagnosis that might come up. This is just a demo view, but that might come up. Secondly, it would identify, flash to the researcher that this is a somatic mutation, kind of not seen in the general population, and you would get the allelic frequency would be calculated and reported. The cause or the impact of that it's a gain of function mutation would also be noted. But it goes actually, the QCI again goes one step further, and not only does it give you the information that I've showed on this slide, identifying a pathogenic nature of that variant, but then, sorry, I didn't realize that I was going to have to go through this, but there we go. Also, it identifies potential associated therapy options and associated clinical trials that might be available. So, for example, here we see that it gives links to the guidelines. And also lights up various clinical trials, and hopefully this is coming through in enough detail that you can see this. But it identifies what phase clinical trials are, where the locations of those clinical trials are. So again, providing a level of detail that isn't found, wouldn't be available to your standard researcher, but again is recommended guidelines. Here we go, treatment capabilities. And so forth. So that's where that panel, kind of not only the front end of what the panel design stems from, but also what the utilization of the QIAGEN knowledge base and QCI as a back end enables that panel design to be used, kind of the end purpose of that as it comes out, and how that enables a greater utilization and greater interpretation of the data that's generated. So now we're going to take a little bit of walk through in the last bit of the slide. We're going to take a walk through or the presentation will walk through the workflow, and then we'll go through some of the data that was generated in a study we did in conjunction with the Broad. So as I said, the panel that we showed was just kind of part of that complete NGS solution. And so if we really look at the NGS solution or the NGS workflow itself, it starts with nucleic acid extraction. You're going to go to a target enrichment, a library preparation, clonal amplification, the sequencing itself, which a lot of people pay perhaps too much attention to, but we have to remember it's just kind of one part of an entire workflow. And then finally, the data analysis interpretation. So, there are obviously QC steps that are built into this variant confirmation. And again, as opposed to some workflows, the workflows that have traditionally been released and are available on the market, where there is no complete solution. Those situations require the individual researcher to assemble various bits and components, some from one manufacturer, some from another. Some may even be kind of home-rolled bioinformatics open source solutions. And those have obviously some challenges associated with them. And we'll go into those in just a little bit. But again, so as I said, we've got this complete workflow. So again, we've got the QIAcube, which is proven effective for your nucleic acid extraction, FFPE, for example. We also then have the target enrichment, which occurs on our QIAgility. The preparation of the library, so taking your amplicons and converting them into the material which will be used in subsequent sequencing, is conducted on the QIAcube. which then goes into next generation sequencing. The clonal amplification stage is or the preparation and amplification is automated also on the GeneReader QIAcube. And then the amplified material is sequenced on the GeneReader. Data analysis occurs as QCI analyze, and then the interpretation, as we mentioned at the last part of the actionable insights section, is done on QCI interpret. However, I think we all have to recognize that there are these QC steps that occur in the process that are necessary for achieving high-quality data, and QIAGEN is able to provide solutions to those as well. So the key steps of quantifying and qualifying the DNA that's been extracted from the FFPE, how much do you have and what quality is it? That can be done through the quantumized system. We then have target enrichment, where we go through QIAxcel to ensure that the material is of the proper concentration, proper quality. And then a similar step can be done on the libraries after they've been generated before they go into emulsion PCR and amplification, which is done also on the QIAxcel. So we cover all of those upstream QC steps. And of course, there's support, and not only are we providing the system itself and the QC steps, but there's a level of support that we're able to provide, and there's a couple of really intriguing features that come along with this, extremely useful. One is the GeneReader System, where the entire workflow and various hints and kind of quick starts are included on an iPad with an app included to kind of a wizard to guide the user through. And then, of course, there's the greater question, which is how do you handle the data, right? And this is where we've seen in the past where it's quite common for people to rely on their homebrew kind of LIMS system, perhaps, or assembled bunch of data management softwares from multiple vendors. But here we provide the GeneRead link, which has that true kind of sampled insight coverage of keeping that continuity of the sample and a LIMS for the data storage at the end of the reaction. So there, I mean, I think I won't jump back because we'll have to go through that whole animation all over again. But one point that I did want to hit was that we're able to provide not only it's significant enough, I think, that QIAGEN provides the entire workflow, but we really went the extra mile to ensure that not only were we providing the quality control steps and the guidance based on those, but obviously the support and the data management systems as well. And then on top of that, you've got access to the QIAGEN technical support and customer support teams who, because every step of the process is a QIAGEN process, are able to provide expert support and guidance on these things. So it's not only are you getting that guidance from the iPad, you can also pick up the phone and get support from trained people. But this is just, and now I'm going to kind of talk about some of the challenges in the workflow itself and some of the other areas that QIAGEN can really bring a solution to this. So, for example, FFPE, I think those of us who have worked with it are aware of the challenges. There's kind of low or at least variable quality and quantity of the DNA, which often depends on the length of time that the material has been fixed, quality of fixation, the type of fixation. All of these things are such that if you're doing a large study, these are variables. Also, there's the challenge of formalin-fixed artifacts, those C to T transversions that come as a result of the fixation, and the fact that frankly, it's a manual process, lots of manual steps. And so by taking this process and automating it as the very first step of the sampled insights workflow, we're able to provide maximized yield and quality for those FFPE steps through this kind of known, people have used this, it's a known, it's respected FFPE sample process. We also include enzymatic steps to correct artifacts that are generated. And the manual process itself, as I said, is no longer manual. It's automated on the QIAcube, which is going to give you a much higher level of reproducibility, consistency, and frankly, reduce that hands-on time and allow the people in your lab to be working on other things rather than just doing extractions. As I said, the GeneReader again we talk about the sequencer to some extent. It's simply part of a connected workflow. there's, as I said before, there's this kind of challenge when you look at the standard workflows that are an ad hoc collection of multiple different vendors and offerings, instruments. One is that lack of a connected upstream workflow, that lack of application flexibility. You may have developed a workflow for yourself that really gives you only access to particular applications. And of course, lack of content with a validated device. And so we're able to address these things. Again, the QIAGEN solution really starts with it's truly integrated. It starts with that FFPE processing itself. Really starts at the core of the issue and takes you all the way through the solution. We give a surprising amount of flexibility as far as the GeneReader itself. We're able to process anywhere from 10 to 4 samples, and those can either be simultaneously. So you can either, if you have 40 samples, you can put them all on the 4 flow cells and run them at the same time. Or, as we know, many lab situations, the sample flow tends to be a lumpy rather than kind of a smooth process. And so you can start, if you have 10 samples that come in, you can start a flow cell with those. Then later, 20 flow cells come in, you can start the next 2 flow cells. And then you can wait and 10 more come in. You can run them in kind of any combination that you like. So it really gives that level of flexibility that I think small labs, medium labs are really going to find very compelling when they don't have that solid base of data that ensures that the machine is going to be run at full capacity every time you turn it on. The lack of content, we've spent a lot of time talking about that. Again, we have that panel that's been designed from the ground up to meet that validated relevance to the subject matter. And then we talk about the downstream bioinformatics solution. Again, we've spent some time talking about the ingenuity and how that not only is that seamless, so there's no user steps required. The run is completed and the data is analyzed. The user is presented with a report that already has a lot of those interpretations built right into it. So we think that this is a real kind of compelling workflow there. We want to dig in a little more onto the bioinformatics solution, which certainly with the acquisition of CLC and their ingenuity pipelines, I think that this is something that maybe we take it for granted and don't give it enough, but kind of enough attention. When we look at bioinformatics itself, certainly in labs I've been in the past, that's been something that simply training people has been a real challenge. But here, kind of that whole time-consuming data analysis can be disconnected from the work that generates it, whereas, and you can get your inefficient data analysis. Pipelines are kind of generalized for all targeted panels rather than specific to the panel at hand. And you really require these kind of super trained gurus, to use these complex tools. And again, the interpretation can be a challenge. And as we've discussed, the ingenuity solution really has a way of turning this time-consuming process into a seamless solution. It's pre-designed. The entire analysis is pre-designed to conduct the data analysis specific to that panel. And regardless of the level of, we've also taken the level of expertise required not only for the analysis but for the interpretation out of the equation, where now the ingenuity is able to give that data in a interpretable level to the researcher, regardless of their level of expertise. I think that both on the typical wet bench area, where I think a lot of the attention gets paid, but also to the extremely important bioinformatics area, QIAGEN has really assembled a solution that provides that kind of complete story here. So now what we're going to do is take a detour into some of the data. This is the result of a study that we did in conjunction with the Broad. And what we were looking to do here was use the Actionable Insights tumor panel on a RAS sample. And what we were interested in doing was generating correlation between our Actionable Insights panel and 2 panels that were actually out that are out in the field already. And we'll walk you through this. So just a quick overview of the study itself. We were using the GeneReader NGS System, some typical kind of QIAGEN reagent pools, and using QCI Analyze and Interpret as the back end. The samples themselves were 43 different colorectal samples. They were FFPE. and they age in years, in sample age from anywhere from 3 to 20 years. And so to say that some of this data was highly fragmented, I think would be an understatement. Some of it was really pretty beaten up. Now, we've shown some data previously. We're using NA128, 78 as the method to take a look at the amplicon coverage. Now here we're using the FFPE sample, so this stuff is really, as I said, extremely beaten up. And what we see is that you do see a dip in the amplicon coverage. So this is the end-to-end amplicon coverage, and we do see that this suffers a bit when you're looking when you've moved from these purified genomic DNA in the NA28, 78 down to the FFPE. You get 97% at greater than 200x and 92% at greater than 500x. But when we look at the insights, and this is what we really care about, is the coverage on the insights. And as I said, these panels are specifically designed to achieve maximal coverage, even in difficult samples, like these fragmented FFPEs. We're getting 99.8% of the variant bases covered at greater than 200x. Excuse me, and greater than 90 or 98.6% of the bases are covered at 500x. So these were really extremely good coverage for those. Very, very impressive considering the nature of the DNA. But that is kind of, to be honest, what they were designed to do. So you get lots of amplicon coverage, lots of over coverage to make sure that you get the necessary insights at the end of the day. Now, here's kind of the interesting thing here is that what we were interested in doing was looking at the TheraScreen assay, which covers these same mutations, and some pyrosequencing assays. And the TheraScreen is a clinical assay. And we wanted to see how well the GeneReader compared to those. And so what we're looking at, using a 5% variant cutoff, kind of the standard. And based on previous TheraScreen and pyrosequencing assays, we were able to determine that 14 of these samples had the mutation, and 29 of the samples were wild type. And when we ran those same samples, but different FFPE slices through the GeneReader system, we were able to get 100% concordance. And so I think this is the first time, to my knowledge, that we've shown concordance between an NGS system and existing assays, IVD assays, in the course of the TheraScreen, but also with the pyrosequencing. So two different orthogonal assays are giving 100% concordance with the NGS system in a real-world situation and challenging sample types, in this case FFPE. So we were extremely encouraged to see this result. So if we dig into this a little further, and we decided to add to the pool comparison with the MiSeq. So this was done again in conjunction with the Broad. The Broad had the samples and did all the GeneReader work and the MiSeq work itself. So if we take a look at the 14 of these samples, the 14 mutants and the specific mutations that they had, what you'll see is that in the left-hand column, you see the TheraScreen assay or the pyrosequencing assay. And in the case where there's a number, that's the frequency of the allele. In which case that you have that indicates that pyrosequencing was used to generate the data. However, because TheraScreen is just a digital or a PCR-based assay, you get an absence or an absence or a presence of the variant. And so in that case, we'll just have a plus in that column, but you don't have any data relative to the exact frequency. And what you see is that at above 5% frequency, we also get 100% concordance between the GeneReader and the MiSeq and the TheraScreen assay. So again, this is just these assays are all kind of arriving at the same number. And so for us, again, just further confirmation of the actionable insights panel. So I'd spent a little time earlier talking about how we were able to do using the QIAGEN offering of the FFPE kit, how that's really designed to use FFPE for the front end, and how that's able to address the formalin fixation artifacts. And so this is a study where we went in and really investigated what is the impact of using this specific FFPE kit. And one thing I want to be perfectly clear, you're going to, just before we get into the data, just so this is first and foremost in people's mind, What we did was we went and we generated samples both using the GeneReader DNA FFPE kit, which has the error correction and all of the kind of benefits of the QIAGEN kit. And then we used the standard sample preparation. Now, these samples were run on both the MiSeq and the GeneReader. And this is not a comparison of the MiSeq and the GeneReader, one to the other. This is rather a comparison of two different FFPE sample preparation processes, which were run on both sequencing systems, simply because we didn't want to find that perhaps we were chasing an artifact that was related to a specific sequencing system. So I just want to be clear about that so people don't take the wrong message out of this particular slide. So if we look at this data, you'll see that with a standard, and we're going to be looking at two different things. One is on the left-hand column, we're going to look at the standard sample preparation versus the GeneRead FFPE kit. And then we'll look at the variants that are identified on the various platforms at either 5% cutoff, a 2.5% cutoff, or a greater than 1% cutoff. Okay, so we're going to kind of lower the sensitivity in three separate increments as we move forward. So if we look at just the basic 5% variant cutoff, you'll see that with the standard sample prep, we get 15, 16 variants identified with the standard sample prep versus 14 variants that are identified with the GeneRead FFPE kit. So those are fairly equivalent. It's really hard to say that there's anything in particular going on there. However, if we drop down to and broaden that window and say we're really interested in variants that are greater than 2.5%, something very interesting happens. So regardless of the sequencing platform you use, be it MiSeq, be it GeneReader, you see that suddenly when you go to 2.5%, the number of mutations that you detect goes up to 25 from 16 from 40. So almost doubles. You're looking at 25 with the standard, 25 regardless of the platform with the standard sample preparation. However, when you do that same analysis using the DNA FFPE kit from QIAGEN, the number doesn't change. You still have 14 variants. So relaxing that window, decreasing that sensitivity cutoff, you still haven't identified any additional variants. And so certainly the concern is that we're looking at these formalin fixation variants that we're beginning to now to see. We're beginning to call them as real variants, even though they aren't. And the problem gets significantly worse if we go down to 1%. Now, it's not reported for the MiSeq, and that has nothing to do with the MiSeq data. It has everything to do with just the analysis software that was used. It doesn't go down that low, so that's an issue with the data analysis software. But if we compare the GeneReader using the standard sample prep versus GeneReader using the DNA FFPE kit, you'll see that you get 38 detected variants at 1% or greater when you use the standard sample preparation. But when you use the artifact correction inherent in the DNA the FFPE kit from GeneRead, you see that you really only add a single variant. So regardless of whether you're looking at it 5% or 2.5%, you get no more additional variants detected, and only a single artifactual variant gets included if you go to 1%. So a really compelling story we feel for the value of using the GeneRead FFPE kit to remove those formalin-associated false positives. So kind of a quick summary here. What I hope I've presented a compelling case that QIAGEN is in generating that sample to insight workflow, that first real comprehensive offering to the scientific community. We're able to offer not only the actionable insights, that the data is generated with an eye towards specifying that the panel and ensuring accurate coverage, high quality data, and truly relevant output through not only the gene panel, but through seamless bioinformatics. But also, we're able to provide that complete workflow where you get a single partner who's able to provide everything from an optimized front end, including FFPE, all the way through under kind of one roof, one manufacturer. So you know that all of the parts have been designed in concert with each other. You don't get an upgrade in one system that hasn't been fully tested with the entire workflow. So you get ease of use for the entire process. You get that efficiency from sample to insight. And you also have the peace of mind knowing that the expertise, at QIAGEN, a company that's known for service, is not only supporting you in implementing, setting up the lab, but also validating and operating that system. And so with that, I guess a little ahead of time, 15 minutes or so for questions. But with that, I'd be happy to answer any questions that people may have. And thank you for your attention.
Thank you, John, for that informative presentation. It is time for our Q&A. If our audience has a question you'd like to ask John, you can do so now. Just click on the green Q&A button at the lower left, type your question into the box that appears on the screen, and hit send. We'll try to answer as many of your questions as we have time for. Now, our first question we have for John: what are the main advantages of the GeneReader system over Illumina or Thermo sequencers?
Let's see. Excellent. Okay, thanks. No, it's a great question. And I think really it comes down to that whole integrated workflow. I think it's a mistake just to talk about it in terms of the sequencers. It's the entire workflow. And unlike either of those two companies, we're able to provide the entire, as I said before, risk of repeating myself, the entire workflow, and that does cover everything from an optimized FFPE extraction, which I don't think either of those two companies provide support for, but all the way through, including the analysis. So I really feel that it's the comprehensive workflow and the support for that QIAGEN is able to offer, and the other companies currently do not.
Great. Thanks for that answer. We do have another question from an attendee. What future panels or content do you have planned for the GeneReader?
Yeah, that's another good question, and it's one that I'm actually going to kind of punt on that one. It's being driven by the market. I think it's also driven by our ability to use the knowledge base and assemble those kind of maximally informative questions that have the broadest appeal. So that'd be a question that I would definitely head towards our sales team. I think they can provide that level of detail.
Good to know. Thank you. We do have another question. An attendee asks, do I need a bioinformatician to run or manage the QCI software?
I can only answer from our experience here in the lab, where certainly I'm capable of running the software. I'm not a trained bioinformatician, so I think it's certainly designed not to require that. I think a lot of labs do. I think that but we obviously don't preclude use by trained bioinformaticians, but it's not required.
Okay, we have another question. Can you please explain more how the 4-flow cells work?
Sure, happy to. So and I guess I'll provide the level of detail that I hope is informative, and please feel free to follow up with another question if I don't address the main concerns. So the GeneReader is built with a turntable. Kind of imagine your old, for those of you old enough to remember records, a turntable at the heart of the system, which enables anywhere from 1 to 4 flow cells to be placed on the system for sequencing. And the system is designed such that if you put 4 flow cells on at once, the run is included in the same amount of time that it takes. There's no additional time required for the flow cells, 4 flow cells. However, what you can do is the unique feature about this is that you are able to start the run with a single flow cell, and it can proceed, however long as the user likes. And then you can actually add anywhere up to the total number of 4 flow cells at that time, kind of in a staggered fashion. So it really allows the users to load the system, load the samples such as they have them, rather than waiting for enough samples to have, for example, to fill all 4 flow cells. So that's, I hope that answers the question. I don't know if there's additional level of detail required. I'd be happy to answer if so.
Thank you, John. We're getting close on time. We have time for one more question. Our question is, what is the total turnaround time for analysis from sample to report?
Question. So again, and just to frame the question, to frame the question here, we're talking about from an FFPE slice all the way out to the report generation. And that the entire process itself is including the seamless bioinformatics, including the sample preparation, library preparation, enrichment, takes 5 days. The sequencing aspect actually, for someone who's kind of been dealing with sequencing as long as I have, it's a little unusual. That's not the greatest part. I mean, it's certainly a component, but you know, the entire process takes 5 days.
Great. That's great. Fast turnaround time. We have no more time for questions. I do want to turn it right back again to John Leamon for any final comments or advice he might have for our attendees.
No advice, I guess. Thanks everybody for their attention. Thank you for the questions. If you have further follow-up questions, I'm assuming that there are ways for you to contact the sales and marketing team, the tech support team here at QIAGEN. And if there are technical questions, obviously I'd be more than happy to have them forwarded on to me, and I will address them as best I can. But again, just thanks everybody for their attention.
Thank you, John. Lots of good information today. I would also like to thank our sponsor, QIAGEN Molecular Diagnostics, for making this presentation possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through the end of August 2016. Attendees will receive an email from LabRoots letting them know when this webcast is available for replay, and we would invite you to share that announcement with your colleagues who might have missed today's live event. That's all for now. Thank you again for joining us, and we hope we see you another time. Goodbye.